Hello, my name is Anniston Anderson. This semester, I had the opportunity to work on research in preparing a CRISPR vector to plan to mutate the AS2 genes in Arabidopsis plants. We used CRISPR technology to focus on the AS2 gene in particular. The purpose of these mutations is to get a better understanding of the protein as well as the structure and the function of these AS2 genes. The CRISPR-Cas9 technology that we use allows us to focus directly on the genes that we are interested in, which in our case again is the asymmetric leaves 2 gene, or what is also known as AS2 in Arabidopsis. Once we target these genes to form mutations, we are able to observe the plants with the mutations for new phenotypes. Since AS2 genes are typically formed to AS1 genes, it is these mutations of AS2 that cause the leaves to not form properly because they cannot form together. This semester, we were able to successfully create a CRISPR vector that will allow us to mutate the AS2 genes. We did this by digesting the pen chimera vector with the enzyme BBSL. Then the photospacer was transformed into bacteria so it could be plated to ensure successful cloning. We then transferred the protospacer into the PDE-Cas9 vector. The final vector was then prepared to insert it into the plants to form these new mutations. So far, we have been able to create and confirm the CRISPR vector and that it will work. We now plan to transform this vector into Arabidopsis plants in order to form new mutations. We will then be able to examine the Arabidopsis plants for new phenotypes and compare them to the originals.